Thanks for coming, everybody. For the Zoom audience, thanks for coming. It's good to see some good friends on the Zoom audience as well. It's a pleasure to introduce Dr. Connor Philson, who has received his PhD in record time, a little over 4 years, being a marmot enthusiast. Connor comes to us from Radford University in Virginia, where as an undergraduate, he thought birds were the whiz, but we know that birds aren't real. He was doing really cool stuff, making technology to sort of, and software to sort of study new questions in bird foraging, and was kind of looking for an ornithology PhD. And then we started talking. He's like, marmots aren't birds, birds aren't real. Maybe I'll study marmots. He has always been interested, as many of you know, in science policy, and I think this has led to real interest in communicating science clearly, into reaching out to different groups, into sort of translating evidence to action, which I think has been a highlight of his career, not necessarily all with marmots, but with other things as well. Today, he's going to tell us about the evolution of social position and structure, a multi-level selection perspective, aka marmot stories you'll enjoy.
Well, thank you very much. Thank you, Dan. Spoiler alert, I'm moving to England and this weather seems apt for the day that we were experiencing. For the people on Zoom, we don't usually get rain in LA, so it's unique what we're having today. So thank you very much for having me. So when we think about the theory of evolution, it is universally celebrated for its explanatory power. So much so that in 1973, the geneticist Theodosius Dobzhansky declared nothing in biology makes sense except in the light of evolution. Now, I don't know about you, but that's a quote that I very much like to use. And the theory was based on a triad of requirements for evolution to occur: variation, replication, and selection. But in Darwin and Russell Walras's time, they didn't understand the mechanisms of replication. It wasn't until the advent of genetics that we had developed a better understanding. But for most of the 20th century, the study of evolution was the study of genetic evolution, in that the only way replication can occur is by genetic inheritance. And this is symbolized no more clearly than etched into the stone floor at the University of Notre Dame, where we have our famous quote around the circle in the DNA helix in the center, as to say, all there is to evolution is DNA. This era is what came to be known as the modern synthesis, or as David Sloan Wilson likes to call it, the Great Constriction. And the Great Constriction, because starting in the 1970s, we began to understand the many types of intergenerational replication, including epigenetics and indirect genetic effects, social and cultural learning in the written language. More modern work from those like Mary Jane West Eberhard and Andy C. have expanded on these ideas with unique perspectives like development and behavioral syndromes. And so going back to Darwin, he thought his theory could explain all aspects of design. Everything that works must have a functional basis. But upon further thought, Darwin came upon a dilemma when thinking about social groups, which is, why do some traits that are disadvantageous on the individual level result in benefits within the context of group living? And so Darwin acknowledged he couldn't explain everything by the product of natural selection alone unless he added something. And that something was to think about natural selection at a slightly larger scale. Writing in The Descent of Man in 1871, Darwin suggested the evolution of group living could affect individual survival. The extension of this today is known as group selection. So here's an example. You got a whole bunch of chickens, and if you're a bird person, and these aren't chickens to you, I'm very sorry I've offended you, but you got a whole bunch of chickens, and you can see that there are really two types of chickens. One chicken is super aggressive and beats up on everybody else, and she lays lots and lots of eggs. The other type is far more pacific and introspective, less aggressive, and has less eggs. And if you put these two types of chickens together, who's going to leave more copies of their genes? Who's going to lay more eggs? The aggressive chicken. But instead, when you have a whole group of the high aggressive individuals together and a whole group of the low aggressive individuals together, what's going to happen is the high aggressive individuals are going to beat up on each other and stress each other into lower fertility rates, as where the low aggressive individuals will get along and lay plenty of eggs and do just fine. And so suddenly we have this very important world in which individual A dominates individual B, but where groups of B dominate groups of A. And this has been a topic of, dare I say, contentious debate since the 1960s. A common way this has been thought about and debated is differential individual survival versus differential group survival, that groups themselves are these entities that are trying to survive. And while this is a case relevant to the natural world in some contexts, it's not the only case where group selection is relevant. In the chicken example, it's not to say that the less aggressive individual is behaving for the good of the group, but that the less aggressive individual is benefited by the type of group that they reside in. Another way of saying this is it's not just who you are that impacts your individual fitness, it's the type of group you live in that also impacts your individual fitness. And so certain group structures can proliferate through time, not for their ability to outcompete other groups, but because of the fitness benefits individuals gain from residing in these groups. Multi-level selection expands on this opposing two-level selection approach of the individual versus the group and proposes selection can occur at any number of levels simultaneously, and where inheritance occurs via the many mechanisms as outlined in the universal Darwinism framework, which may run contrary to some popular books in our field. And these multiple levels could be cells succeeding at the expense of other cells within the same organism. Perhaps one way to think of that is cancer, or genes cooperating with one another where selection then occurs at the level of the individual organism, which is perhaps how we traditionally think and teach about this. And on the group level, it could be microbiomes to entire cultures and ecosystems. Multi-level selection is a framework that incorporates all of this by saying natural selection can occur simultaneously on multiple phenotypic levels relative to each other and potentially in aligning or opposing directions. And so multi-level selection may be a relevant and important evolutionary mechanism. And let's be specific with exactly what we're talking about here. Selection and evolution are different. Natural selection acts on phenotypes regardless of the genetic basis, whereas evolution is the genetic response to this selection. And so selection is a necessary prerequisite for evolution. So where we find selection for traits at multiple levels, we could then find evolution for these traits at multiple levels. And multi-level selection has already been documented as a driving force in the emergence of the first multicellular organisms to the structure of entire ecosystems. It also acts on morphological phenotypes in plants and reproductive strategies in animals. However, despite social behavior being the common example in making the case for multi-level selection, no work has explored the presence and strength of multi-level selection for both individual and group social behaviors in the wild. And so in the absence of multi-level evidence for multi-level selection for social behavior in the wild, this decades-old debate going all the way back to Darwin's The Descent of Man of individual versus group selection remains largely a theoretical and not analytical debate. And this question has the potential to reshape our understanding of the evolutionary origins of friendship and social organization that underpin our social lives and social lives across species and underpin our societal institutions across governments and across nations. And social behavior is a particularly interesting phenotype to ask this question because from social group living, we can ask about selection for where an individual is positioned within their group and the overall structure of their group. These are both byproducts of group living. Not all individuals are as social as others, and not all groups are the same. An individual's position depends on their own behavior, but also that of others, whereas the group phenotype is an emergent property, more complex than the sum of its individual behaviors. Thus, these are two distinct levels of biological organization that have potentially different consequences and are under potentially different selection. And as we thought about with our chickens example, this is demonstrated where the non-aggressive chickens experience individual fitness costs from their individual phenotype, from their social position, but experience individual fitness benefits from the group phenotype, the type of group that they reside in. And we can measure these two levels precisely with social network analysis. Networks are a simple way to represent complex behaviors. I often like to joke that it's just drawing lines between dots, where the dots are individuals and the lines are social interactions. And with network analysis, we can ask not just how many social partners you have, but how frequent are those social interactions? Which of your connections are connected to each other? And your position within the larger group, incorporating both your direct and your indirect social partners. And for social structure of the group, we can ask how reciprocal is the group? How polarized? How breakable? How homogeneous? How connected or sparsely connected your group may be? And so with social network analysis, we're able to quantify the many unique aspects of social behaviors on multiple discrete levels of biological organization to ask not just how your own social behavior impacts you, but how the type of group you reside in impacts you. And I want to make this very clear. At both levels, we're talking about the fitness consequences to the individual. We're interested in how social individuals are because that affects them, and then how social the group that they reside in is and how that impacts them. We're not talking about group proliferation, groups competing with other groups. That's a talk for a different time. But if there is to be evolution of social position and social structure, we must first complete our Darwinian triad of variation, replication, and selection. But social position and structure, like most biological traits, are not completely independent, and we'll get to this. But if we are to clearly understand their evolution, we must understand their selection concurrently and relative to each other. So if we modify Darwin's triad just a little bit, we're looking for variation, replication, and multi-level selection. So I'm going to tell you a story today about marmots in 3 core parts: the drivers of variation, the consequences of this variation at 2 levels, and then lastly, we'll put it all together to estimate the relative strength of multi-level selection. And throughout the talk, we'll come back and check on our progress on our Darwinian triad, because if we have these 3, only then would evolution be possible. So hopefully you're asking, what the heck even is a marmot? So they're large ground squirrels in the rodent family. There's about 15 species in all. Some species live alone, some live together. Here in the United States, we have a national holiday for marmots. So make sure you celebrate in February. Yes, groundhogs for you on the East Coast are Marmota. But the marmots that we study are the yellow-bellied marmots, Marmota flaviventer. Hereby, I'll just call them marmots. They're harem polygynous, where one male tries to control multiple adult females for whom the social groups are formed around. They're obligate winter hibernators, and they're facultatively social, meaning they're found in a wide range of group sizes, sometimes alone. And the marmot annual cycle is essentially wake up, reproduce, get fat, go back to sleep. Yearlings disperse, so there's this life history decision that has to happen when you're a yearling, but this is generally the life cycle for all the individuals. And so really it is a game of get fat or die. And get fat or die is so important, and when I finally made every kid's dream come true, this is what I told them for some reason in Nat Geo. This is what I told Nat Geo and got quoted. They also quoted me thanking the marmot for not pooping all over us, so clearly I've made somebody mad over Nat Geo. And we study the population at the Rocky Mountain Biological Laboratory, which is this incredible and truly unique biological field station in Western Colorado, where we've been studying them annually since 1962. So in the last 60 plus years, we have over 4,500 uniquely marked individuals, over 200,000 behavioral observations, and over 23,000 hours of observation just since 2002 alone. And our marmots live in colonies in 2 primary areas, a lower elevation and a higher elevation site. The higher elevation sites experience harsher weather conditions comparatively, and winter lasts about 2 weeks longer up at those higher elevation sites. And this makes it the 2nd longest running project on free living and wild mammals in the world. It's second only to Jane Goodall. And if I'm going to come second to anybody, Jane Goodall is the best person to come second to. So for 5 months a year, we go out 6 days a week, often twice a day, to observe and trap these animals. During this, we mark all the individuals and take a suite of physiological, hormonal, health, and genetic data for analysis, and a suite of behavioral data. So we know who's where, who's interacting with who's initiating those interactions, and who's receiving those interactions. We also know what predators are around and the environmental conditions. And these marmots display a wide variety of social behaviors, from sitting together looking for predators, to greeting, to allo grooming, to foraging together, to playful biting, and to all out pouncing on each other. And so with this variety of behaviors, we can construct their social networks. The networks I'll talk about today are all affiliative, which represents some 88% of all of their social behaviors. We also can build agonistic networks for that remaining 12%. Those are more sparse, and we use those to ask additional questions. So today, we're focusing on affiliative play behavior. The networks we build include yearlings and adults and are based on space use overlap and social interactions. And of course, in a 60+ year project, all the questions I'm asking today wouldn't be possible without hundreds of people before me, like Ken Armitage, who started the project in 1962, and Julian Martin, who co-PIs the project currently, and Dan Blumstein, who's the project PI and my PhD advisor. Some of the work I'm talking about today will come from this incredible group of people, including 5 undergraduates I've had the honor of mentoring and working with, whose work we're featured today as well. So that's the system. Lots of data, uniquely marked individuals, known genealogies. We know a good amount about their biology and seasonal patterns and the variety of social behaviors that they display. So what about the drivers of this variation? And we're interested in the drivers of individual variation, specifically the genetic and environmental drivers, because it is they that generate the social phenotype, which could then be under selection. And before we get to variation in social behavior, first, why do marmots live in social groups in the first place? Social position and social structure are, of course, byproducts of this group living. Well, likely to avoid predators. Marmots are a tasty snack to a range of predators, from birds of prey to foxes and badgers. And it's really a numbers game for the marmots. The more bodies around dilutes the probability of getting eaten. The more eyes looking for predators and alarm calling at predators increases the probability of escaping safely. And so marmots live in these social groups, and these groups, even in the same place, vary from year to year. And these are really small examples of marmot social groups. They can get as large as 30 individuals, but that is less common, and this is much easier to visualize. And so with these different group sizes comes different patterns of individual sociality. And so individuals may have preferential relationships with others. They may be more or less isolatable to others, or more or less connected. But what are the drivers of some of this social variation? We know that larger habitat patches have larger group sizes, and that marmots become less social as that group size increases. Marmots are more friendly to others in the same age class and to closer relatives. And perhaps unsurprisingly, like us, marmots become less social as they age. So demographics and location matter. But what about the abiotic environmental drivers? And given marmots are a seasonal hibernator living in the high alpine, the seasons, they're stark and they're pronounced. And thus we differentiate between weather, like precipitation and temperature and humidity, and the timing and duration of the seasons. And this work was done with an incredible undergraduate student here at UCLA, Carla. And we found 3 key associations when exploring both weather and seasonal timing's relationship, social position. All 3 of these relationships were with clustering coefficient, which measures the extent an individual is clustered with others. It's the clickiness of an individual. And so as we move up our y-axis here, we have more social connectivity. And we know clustering coefficient is important for the structure of social groups. So these results could matter across our scales, across our 2 phenotypic levels. And clustering coefficient was associated with 3 key environmental variables. The length of the previous growing season, the length of the growing season before hibernation, which is a proxy for how much time they had to get fat. The length of winter, which as longer winters may require more body mass and may shorten subsequent growing season. I'm jumping the gun here. And so, we also have 50% snowmelt date, which is the timing where we consider winter to have ended and the growing season to have begun. As 50% of the snow is melted, there's now bare ground for the marmots to begin to eat. And so we found a positive relationship between social connectivity and growing season's length. We also found a positive relationship between social connectivity and winter's length, but we found a negative relationship between social connectivity and the timing of winter's end / spring's beginning. And so it's the duration and timing of seasons that relate to marmot social position in this system. And given climate change is making abiotic environmental conditions more stochastic and is driving mismatches in seasonal timing and phenology, this is an important relationship to understand, and there's much more work to be done here, especially within this system, as these results we could spend the rest of the time unpacking on our own and across all taxa. So what about some of the biotic environmental drivers? We were interested in how the diversity and composition of the gut microbiome and how that relates to social position. And given extensive work in this field with a focus on laboratory systems, this could be an important link explaining some variation in social position in the wild. And this work was also led by a rockstar undergraduate, Madison. And on our y-axis, again, we have clustering coefficient, and on our other, we have embeddedness, which is how central or connected an individual is within their group. More embedded individuals would be harder to break out, to remove from their group. And on our y-axis, we have streptococcus, which is related to strep throat in humans. And we found that greater abundance of this strep throat related bacteria is associated with decreases in social connectivity and social position. In other words, more sick marmots may be less central and connected within their groups, which I think we can all relate with. And so we know some of the environmental drivers, but is there a genetic component to the social phenotype in this system? Lay et al. In 2010 used a quantitative genetics approach, the animal model, to estimate the additive genetic variance of social network traits. In other words, are these individual social network traits heritable? And the answer is yes. Genetics alone explains 20% of some social traits. Another way of saying this is that marmots do have some genetic predisposition to their social position. And so this genetic component, plus our environmental component, gives us insight to how the social phenotype is generated. But what about the fitness consequences of the social phenotype? And why we won't measure fitness directly in this talk today, i.e. gene contributions to the population, we instead use common fitness correlates like reproductive success and survival. And so quickly coming back to our Darwinian triad, we have variation and we have replication. So we're building our case towards evolution. So let's move to explore the consequences of this variation at two levels. Since 2009, the fitness consequences of social position have been explored in a variety of ways, all with social network analysis to illuminate the costs and the benefits of social position. And so the results I'll show you here is that as we move along our x-axis, we're assuming that increased sociality would lead to increased fitness. So all the results will be for what are the consequences and the benefits of increased sociality. So we know that more social females have fewer kids, and more social individuals are more likely to die over winter, and that more social individuals have shorter lifespans. On the benefit side, we know more social adult females have increased summer survival, and more social yearlings are less likely to disperse. And dispersal could be a costly behavior to engage in, crossing rivers, climbing over mountains, exposing yourselves to predators. And so these are all done with a variety of social network measures, but generally social connectivity is costly. And the dispersal finding tells us that social position is not only influencing fitness consequences, but these major life history decisions as well. Working with Tally, who was a student I mentored out at Rumble this summer, we were interested in another decision, when to flee from an approaching threat. The social security hypothesis predicts that more socially connected individuals would perceive less fear and thus flee from a threat at a shorter distance. So this would be you're only fleeing from the predator or the threat when it's on top of you versus further away from you. And this is especially relevant because if marmot group living is all about predator avoidance, how connected you are within your social group should matter. And it does. Individuals with more social interactions perceive less risk. Now, this is a very modest relationship. I'm going to stress the word modest here, but it is statistically significant after controlling for all the other biologically relevant variables that we fit in our models. And so hopefully you're thinking, that's a lot of costs to increase social connectedness, and it is. Marmots are relatively unique in the consequences of social position. Most species, kind of like the rhesus macaque, experience mostly benefits to increase connectivity within their social groups. Marmots are the outlier here. Perhaps this is attributable to their facultatively social nature, as other species here are mostly obligates in terms of their social behavior. They're more gregarious than our marmots. So from all of this, we know some of the drivers of sociality, and we know that increased sociality is mostly costly in this system. Marmots want to go to the party, but they want to stand in the corner and not really interact with anybody. They want to go to the research seminar, but they want to sit way in the back. Marmots largely want to live in social groups, but they want to avoid predators and not be all, they want to live in groups to avoid predators, but they don't want to be all that social within the groups. And so that's the individual level. And if that's mostly costly, perhaps there are benefits from the type of group and the structure of the group that you reside in, the group social phenotype. And so earlier we explored the drivers of social variation, but largely on the individual level. And hopefully that left you asking about drivers of variation on the group level. We know from the universal Darwinism revolution that the units of replication, how groups are inherited, how group traits are inherited, need not be genetic, but instead are likely a mix of genetics and indirect genetic effects and cultural transmission. This is an area much in need of research, but in 2021, Cantor et al. Began to lay the conceptual framework for the structural feedback between individuals and their group. In our marmot system, even the removal, especially of a single adult male, can have global consequences on group structure. So as you mathematically knock out a single individual, the structure of the entire group can change. So there's clearly a feedback here. But with social network analysis, there's a whole suite of measures we can use to quantify the group in a way more complex and perhaps emergent from the individual level. Now, each of these measures tells a different biologically relevant story that we can develop a priori predictions for. But broadly, these 6 measures can be categorized into 3 core groups: the connectivity of the group, the homophily, similarity of the group, and the shape of that group. Density, for example, tells us what proportion of connections are observed out of all possible connections. Cut points is how easy it is to break that group into 2 or more social groups of at least 2 individuals. And degree assortativity is kind of like extroverts hanging out with extroverts or introverts hanging out with introverts, or if you have these unassorted groups of all sorts of different types of individuals. And so we predicted that largely there would be negative consequences of increased connectivity, given that connectivity is costly on the individual level. But we predicted largely benefits from increased connectivity of your group, given more similar social profiles may reduce costs. And we predicted a negative relationship for shape, to say that the star network is likely to be centered around a single adult male or adult female who may be aggressive and beat up on others. So a more centralized group around one individual may be more costly than a decentralized group. And I normalize all of these measures by group size to control for that effect that group size has on these measures themselves, because the larger the group, the less likely it is to be connected. And so we account for that by controlling for group size. And studies in humans have already begun to use social structure in these measures we just talked about to understand patterns of efficiency in office workplaces, or explore the attributes that lead to higher winning percentages in online team-based video games, or the vitality markets, the vitality of housing markets in China. These measures have even been used to quantify social structure via the efficiency, or perhaps better said, the inefficiency of the US Congress. And so with these global social network measures, we can ask precisely, not just about how your own social behavior impacts you, but also about how the type of group you reside in impacts you. Because if these 2 levels have different fitness consequences, then we know that the individual and the group social phenotypes, while not completely independent, are indeed quantifiably discrete. And if the consequences of these 2 levels are different, it will be easier to identify a potential multi-level selection. Otherwise, it could be interpreted as just individual level selection expressing itself at 2 different phenotypic levels. And so, looking at reproductive success, on our y-axis, we have the number of offspring weaned, the number of cute little baby marmots that emerged from the burrow, and this is just for adult females. It's much harder to generate this data for adult males. And on our x-axis, we have that cut points measure, which tells us how many social relationships there are that have broken or lost due to death or immigration, result in 2 separate social groups. So as we move down our x-axis, we have more easily broken groups. And so actually, as we move down our x-axis, groups are less connected. And we found that females who live in socially connected groups have less kids. This is a very modest association, but shows some attributes of the group might influence fitness, even after we control for other biologically relevant variables like body condition, location, age, individual ID, and year. Here we have all of our measures of social structure on our y-axis or on our x-axis, and y we have our summer survival. And this shows that there is no relationship between summer survival and any of our measures of group social structure. Perhaps this is surprising given the benefits of social connectivity on the individual level for summer survival, and how predator avoidance is likely driver of marmot social group living, and that predation accounts for 96% of summer mortality in the system. So the lack of result here is interesting in its own right. Looking at winter survival, we again have cut points on our x-axis, which is how easily the group can break into two or more social groups, with groups being less connected as we move down our x-axis. And we see that marmots in more breakable groups are less likely to survive hibernation, or in the reverse, more connected groups have higher individual hibernation survival. And this is surprising for two reasons. One, the relationship on the individual level for social connectivity is in the opposite direction when we measure social position. And we just observed the opposite direction of a relationship for reproductive success and cut points. Again, I'm going to stress the word modest, but statistically significant relationship suggesting that group social structure matters in different ways, depending on the fitness context and after accounting for other biologically relevant variables. And while this isn't one of our fitness correlates of focus today, I wanted to shout out the incredible work of an undergraduate from a few years ago I mentored both here at UCLA and in the field that found residing in more social and connected groups is associated with decreased rates of mass gain. And if you remember how important get fat or die is, this bolsters the importance of group social structure for individuals. Lastly, Sarah, another undergraduate from Rumble this summer, we took the social cohesion hypothesis, which says more social individuals are less likely to disperse, and extended that to the group level to ask if living and residing in more connected group structures results in less individual dispersal. And we found that individuals residing in groups that are structured around one or few individuals resulted in more dispersal. Again, in a network that would look like this, it's very likely that that central individual is an adult female or adult male, potentially beating up on others. And so this is a case, if you live and reside in this type of group, you're more likely to disperse. And so not only is the type of group you reside in impacting your individual fitness, but also these major life history decisions as well. And so we have fitness consequences at both the individual and the group levels. Both the individual and group social phenotypes have consequences. And despite individuals structuring groups, and thus the two groups not being, the two levels, these two phenotypes not being completely independent, we observe different individual fitness consequences between these two phenotypic levels. And so these two levels of biological organization may indeed be experiencing multi-level selection, and potentially with different directions and magnitudes of this selection. So again, we have variation and we have replication. And so for our last component of the triad, we must explore social position and group social structure together in a unified framework. By exploring these two levels in the same model, we can assess the independent contributions of the individual and group social phenotypes to individual fitness. In other words, do we have multi-level selection? And let's remind ourselves with exactly what we're looking at here. We're asking about selection, which is independent of any genetic basis. And we're asking about selection acting on individual fitness variation at both levels. We're not talking about differential group success. Again, that is a talk for a different time. And so to aid our exploration of multi-level selection, we chose 4 pairs of analogous social network measures that tell a similar story across the two levels, such as how isolatable an individual is within their cluster and how breakable their social group might be. And these are all measures that we've used in the previous analyses I've shown you today. And you could be asking, do we need these analogous pairs for multi-level selection? No, most certainly not. But it does help strengthen the case for multi-level selection and simplifies the interpretation if we do find selection for these pairs within one of these pairs. Because perhaps we could find connectivity, for example, is under positive selection at the individual level, but negative selection at the group level, or is just under selection at the group level, but not the individual level. And so we fit these 4 pairs of analogous traits into a contextual analysis. And that contextual analysis allows us to partition selection among the two levels relative to each other. Contextual analysis is an extension of the Land-Arnold selection analysis using partial regression to partition selection among phenotypic levels. In other words, it's really just a fancy linear model. And it's fancy because it requires the traits that we're interested in to be scaled in a biologically relevant and interpretable way in order to interpret the selection gradients. And so here we have our basic structure of our contextual analysis, where our dependent variable is going to be a fitness correlate, say an individual fitness correlate, say summer survival. And then we have our measures of our individual social phenotype and our group social phenotype. And for the individual social phenotype, we scaled those within groups, within years. And for measures of our group social phenotype, we scaled those across all groups, across all years. And so we have three separate suites of this contextual analysis for three different measures within each suite of individual fitness: summer survival, winter survival, and reproductive success. For each measure of fitness, we additionally fit other biologically relevant variables, such as predation for the summer survival models, or fall body mass for the winter hibernation survival models. Each model also had valley location, remember our higher and our lower elevation sites, and social group size, because we know that's important for all of this, as additional fixed effects. And within each suite of these contextual analyses, we broke the analysis down by age, sex class: adult female, yearling female, adult male, yearling male, because these four different classes, these four cohorts, have fundamentally different life history strategies. And so to account for that, we broke them down within each contextual analysis suite. And so what did we find? Well, I know there's a lot here, but multi-level selection is happening. So let me walk you through this. On our y-axis, we have our different measures of individual fitness. These four here are for hibernation survival, and these two are whether or not a female weaned any offspring, one, a dozen, does not matter. Did you wean any offspring that year? On the x-axis, we have our different measures of the social phenotype. These two are the individual, these three are the group. So again, significant individual traits are in green, significant group traits are in blue. There's some positive selection, there's some negative selection. 4 of the 6 traits here we found that were under selection was in adult females. And given marmots, social groups are structured and they're matrilineal, so they're structured around one or few adult females. This is particularly interesting and not all that surprising, perhaps. And what this shows is that selection for the individual social phenotype for female reproductive success and selection for the group social phenotype for winter survival across age sex cohorts. And because these two levels of the social phenotype are scaled, we can compare the strength and the magnitude of this selection with the estimates. And so the mean strength of selection for the individual phenotype is 0.76, as where the mean strength of selection for the group phenotype is 1.03, which suggests selection is stronger for the group social phenotype. Now, we didn't find a case for multi-level selection happening within any of the analogous pairs or within the same fitness age sex settings, suggesting that traits at one level are also not under selection at another level, but that some traits are under selection at the individual level and different traits are under selection at the group level within each of these age sex groups. What I find interesting, one of the many things I find interesting at all of this is that, again, summer survival wasn't under selection at either level. And remember, selection is for group living in this system is likely predator avoidance. And so this could potentially suggest that the selection for group living is different than the selection for the subsequent individual social position and group social structure, which are, of course, byproducts of that group living. And so all of this says is that how social an individual is and the structure of their social group are both under natural multi-level selection. Now, a common argument against group selection and thus multi-level selection that has been used since the 1960s focuses on differences in individual lifespan versus the group's duration. Where individual lifespans are shorter than group duration, theory suggests selection proceeds faster at the individual level. However, in our marmots, dispersal, mortality, births, and hibernation ensure that groups restructure annually, and individuals surviving to adulthood have a mean lifespan of 4.07, and thus are part of multiple social groups within their lifetime. And because individual lifespan is shorter than group duration in our population, This traditional argument from the 60s does not apply. Indeed, the same logic, but in reverse, would predict stronger selection at the level of the group, which is indeed what we found. So we have variation for social position and structure. We have replication for social position and structure, and we have multi-level selection for social position and structure. And so we've completed Darwin's triad, and the evolution of social position and structure is indeed possible with this multi-level selection perspective. But of course, for evolution to occur, we need intergenerational changes in the units of replication. And given that individuals are genetic and groups are a product of multiple units of replication from the individuals who comprise that group's genes to indirect genetic effects and cultural transmission and learning, we can both be confident that natural multi-level selection is happening and acting on social position and structure, and say that more work is needed to be done to clearly understand if and how evolution for both social position and structure is occurring. We need to understand how groups and group structures form and are maintained. What specifically are the units of intergenerational replication for these group structures? We need to understand these two phenotypic levels across time and across space. We need to understand multi-level selection across species. And of course, we need to understand if there are changes in the intergenerational units of replication in response to this selection, whether that be genetic or non-genetic on the genetic or non-genetic levels. There is a lot more work to be done here. This is why in January, I'm joining Dr. Lauren Brent as a postdoctoral fellow at the Center for Research in Animal Behavior at the University of Exeter in England, where I will continue to explore these questions with two powerful data sets. Rhesus macaques on Cayo Santiago in Puerto Rico. I mean, that doesn't look so shabby. I'll go there. Where there have been free living, but not wild, right? There are no natural predators and these animals are provisioned, so they're free living, they form their own social groups, but they do receive food every day. And that population has been studied since 1932. If you're curious on how they got there, we can talk about that later. Allowing me, and this population allows me to ask detailed questions about how groups form and how they're structured, about multi-level selection, and about social evolution, both genetic and non-genetic, given there's an incredible genetic and behavioral data set on these animals. And if you watched 60 Minutes 2 weeks ago, you learned all about this incredible system. And if you haven't watched 60 Minutes, be old like me, go watch 60 Minutes. I'll also leverage the MacaqueNet database, which includes social interaction data across a dozen species of macaque, to ask comparative questions about multi-level selection and social behavior and how social structures form and look different across all these species. And of course, I'll be continuing my work with the Rumble Marmot Project to continue to teach and mentor in this incredible biological field station and to ask more questions about multi-level selection and potential genetic and non-genetic evolution in response to the selection that we just saw today. So it's not lost on me that I've thrown quite a lot at you. So in simple and in pointed terms, what did we learn? We learned that all sorts of things drive variation in social behavior, like the microbiome and seasonal timing. We know that social behavior is genetically heritable and that both these genetics and environmental drivers produce the social phenotype. We learned that these social phenotypes have fitness consequences at two levels: individual social position and group social structure. We learned that not only is multi-level selection happening, but that multi-level selection can be stronger than individual selection alone. We learned that we have our Darwinian triad of variation, replication, and multi-level selection. So hopefully you're asking, is there evolution for social position and structure? I'm going to leave you with that question, and I look forward to coming back one day soon with answers. Now, of course, there are too many people to thank. This is a very non-exhaustive list, but I want to be sure to give Dan a big thank you for his mentorship in keeping the Rumble Marmot Project alive. Long-term research projects like this are vital to our understanding of ecology and evolutionary biology. And I also want to thank Rumble, especially the staff that make research like this possible in the same place year in and year out. And they make it possible, they make it accessible, they make it enjoyable, and the community at Rumble for inspiration and support. And thank you all very much for coming and having me both today and over the last 4 years. And I'm eager for your questions. Michael, you're right here in front.
Yeah, great talk, Connor. What can you speculate about the sort of drivers of evolution of group social structure when you look sort of comparatively across roles? Do you get any insight into you know how often we see the sorts of social groups that you find in marmots and other groups and when you're looking at more asocial groups that sort of yeah, does any of this sort of tie in with your ideas of why community organization evolves and why it is also why you see the diversity of community groups that you see in marmots?
I mean, social group living is always a numbers game, whether it be to avoid predators or find resources or penguins huddling together for warmth. But the marmots are relatively largely unique because they're facultatively social. They experience mostly costs. So they're not meerkats, right? They're not social birds. Sorry for the bird people. They're weird, right? And so I would expect, while most people haven't done this work yet, that the consequences, the largely negative consequences of individual position and structure we see in the marmots would actually be more connectivity would be good in most other species. We see that in blue tits in England. We see that in some primates, the Cayo system actually in Puerto Rico. So marmots are definitely a bit of an outlier in terms of not why they live in groups, but the consequences of how social they are within those groups once they live in the groups. So marmots might be the outlier from a comparative evolution perspective.
In your networks, how explicitly were you accounting for relatedness? Because I would think that would be the single greatest predictor of any kind of group change. And I was surprised that you didn't really invoke inclusive fitness forward, and a lot of the response variables were very individual fitness based rather than.
Yeah, I'm looking at Peter here laughing in the back as we've had this conversation many times before. The marmots, they're matrilineal. And so an adult female kind of runs that social group that we look at, and they recruit their offspring to stay. So there's really high relatedness in these social groups, and that won't vary too much from social group to social group because these groups are matrilineal. Those matrilines track through time. We could go back and we could look at these pedigrees and follow the matrilines through time, but they're relatively stable. So there's really high genetic relatedness. There's also, for people far smarter than me, who break down the math between looking at inclusive fitness and kin selection versus multi-level selection at a certain level of high relatedness. Some folks argue, some folks don't argue, that there is mathematical equivalence between those models. So you could, depending what camp you're in, interpret all of this both as, you know, a kin selection of inclusive fitness versus just a multi-level selection perspective. I'll leave that to the maths people.
I'm curious. I think I would say like a lot of the arguments against group selection, you know, are arguments against sort of genetic evolution. And I'm curious, you know, how much of the genetic evolution is happening, because you know these effects might be so modest, these population sizes might be so small, and you know if these social traits themselves are not super variable, it seems to me like there might not be much genetic effect. Like all these social groups are just so transient.
So we haven't started looking at that yet. Sadly, I'm leaving. I'll come back. I'll do more. That's the question we want to ask, is on the individual level, specifically about the genes and the group. We'll have to integrate how we think about genes and groups and indirect genetic effects, all those universal Darwinism levels of thinking. Without having looked at it in full, there's potentially selection, but not subsequent evolution, which would be, I think, very interesting on asking why that might be. Why are we seeing selection, but no genetic response, no evolution? So I don't have that answer for you. It's a curious answer. I need to learn a lot about genes in my future. But there could be a case, and it might be the case, that there's no subsequent evolution, no genetic response. Dave.
Given that things aren't changing necessarily much, and might be hard to study evolution in that consequently, is there any role for manipulation that you could envision to address the evolutionary question?
So we had the paper with the mathematical knockouts. So we won't do experiments with these animals. We've studied them for almost 63 years now. We won't go and hand remove individuals. Some people do that. They remove birds and keep birds in a cage, usually to do social learning and watch transmission when they put them back in the group. So we're not going to do that with the marmot system. I don't believe they do that in Puerto Rico, but you can do it mathematically. You can do those topological knockouts where you simulate the networks with all the data you have and then remove individuals. So while the math approach isn't the best approach necessarily, we're not necessarily going to do that in the field as not to disturb these wild and free living animals. But I wish. Another question?
Given that there are consequences of having a certain degree of sociality, what level of connectedness, what are the consequences of those consequences? In other words, do marmots have a certain level of connectedness? How does that affect social structure?
Yeah. Is there an optimal group size to marmot living? Well, we know that as those habitat patches increase the social, you can fit more marmots in the same amount of space. And so there is likely some aspect to ideal group size based on where they're living. The more predators that are around, so sometimes foxes will dig their burrows right next to the marmots and live and set up shop and then just pick off all the baby marmots. And so maybe that's a case where your optimal group size might shift. So I'm going to say that's largely dependent on environmental factors, less so perhaps than just the social factors.
Yeah, so similarly to the inclusive fitness argument, obviously the number of offspring that you have has a profound impact on the group structure that you emerge into every year. And I was wondering, like, is there any control to that? I mean, as in, like, because that's sort of a very much like a fitness on the group structure argument that doesn't invoke like any sort of prior group structure, right? As in, like, the reproduction directly causes the structure of the group rather than the consequence of it. So are you controlled to that? And if so, how?
So I controlled all for group size. When you look at some of these group measures, you can also control for the density, right? The proportion of all those interactions, because we know at these larger levels that density matters a lot as well. Our data is not as a high enough resolution. The grass grows very quickly after the snow melts, and then the babies are yay big. And so it's really hard to see all and how the networks might change after you have that influx of individuals halfway through the year. So yes, you could. I don't know if we necessarily could do that now, but that's most certainly a question we're going to keep in mind.
A question on Zoom. Do you think the strength of the individual and group selection trends we've seen here are different, perhaps weaker, now since climate change and multiple systems led to greater interannual variability and seasonality? The last few generations of marmots probably been experiencing greater fluctuations in the one species.
So I won't repeat that on Zoom because I assume everybody on Zoom can see the chat. Yes, so the question is, how does seasonality and shifting phenologies influence this social behavior? Well, luckily we have a graduate student who's looking at these things sitting in the back who read the question, who's going to break down those questions about how seasons and weather influences social behavior. So I'm going to have to deflect that one 2 or 3 years down the line. You're going to have to come to her talk in a few years. Well, I don't want to hold people. So thank you very much for having me.